Okay, so we're going to do 10 minutes on microscopy, generally on the required practical rather than the theory, but some of the theory as well. So I'm going to stop my timer. Oh, reset it. Okay, let's go. So, microscopy. So this is pretty much the things on the required practical that I'm going to put on there. So I'm going to start with preparing a slide. I'll move this over so I can write a bit more easily. So this is where we're preparing a slide to use underneath the microscope. And usually we'd be asked for either a cheek cell or an onion cell. So this is a kind of slide like this. So it's how do we prepare that slide? So I'm going to start with, I'm going to do onion cell as my first example. So I'm going to use tweezers. Put a thin layer of onion epidermis. Off the onion. I'm going to place that on a glass slide. I'm going to lay it flat. So I'm just making kind of notes here. Then I'm going to add a drop of stain. And because this is a plant cell, the stain that I'm going to use is iodine. And it's important to know that. So I've got some keywords coming up here. I'm going to use tweezers, I've got a glass slide, I'm going to use iodine. These are all important things that I need to know for that methodology. Then I'm going to carefully place a cover slip. That's this really thin piece of glass that goes over the top. So this carefully place a cover slip. I'm going to do this at an angle to avoid bubbles. And I'm going to blot away any excess stain. So I'm going to place a cover slip, avoid bubbles, and a blot excess stain. So a good question is why are we using stain? And that's to help visualize the cells. Okay, so this one, I've done this here. This is all with onion. If I want to do it with cheek, I'm going to use a swab. So a cotton swab. To rub that inside of the cheek. I'm going to wipe that onto the slide. And then I'm going to dispose of swab safely, thinking about biohazards there. And then the stain I'm going to use is different. I'm going to use methyl blue. So it's good to have different stains. So this time it's slightly different. I'm going to use the cotton swab and I'm going to use methyl blue for the cheek one as well. Okay, so let's have a look. So I've got all of that. Then I'm going to need to take my slide and use it under a microscope. So this is a microscope. But one of the things that I need to know is the different bits of the microscope. So next to this video or somewhere on my website, you can actually just print this picture out because I'm not going to waste time drawing a microscope because I don't need to draw a microscope. It's not something that I'm going to be assessed on and I'm not looking at my art, but I want to be able to label the different bits of it. So this is a light microscope and here you're going to have your eyepiece lens. So this is the bit that you look down. Here are your objective lens. And there's 3 of those and they have different magnifications. Often they're 4 times, 10 times, and 40 times, but they might be different ones. And these rotate around here, so you use one of these at a time. Your eyepiece lens often has magnification 10 times as well. So one of the things to remember is that when you are looking down a microscope, you're looking through the eyepiece lens and then down through the objective lens as well, which means that your total magnification is equal to your eyepiece lens times or multiplied by your objective lens. So for example, if your eyepiece was 10 and your objective lens was 40, your total magnification would be 400 times. So we've got in here, the first thing that you need to remember in terms of calculations to do with microscopy, your total magnification is your eyepiece lens times your objective lens. Might just make this stand out a little bit here. Okay, then you've got your stage. You've got your coarse focus knob, and you'll have a light source here as well. So your stage is where your specimen is put. So those are all the different pieces of the microscope that you need to know. Okay, so we also need another method for viewing the specimen. So once we've made our slide using this method and we know about our microscope, how are we going to actually view that slide underneath the microscope? So the first thing that we are going to do is place the slide on the stage. Then we select the lowest power objective lens. Then we're going to use this coarse adjustment to move the stage to just below the objective lens. We're going to move this stage right up to just below the objective lens. So use coarse adjustment. Use coarse adjustment to move the stage to just below the objective lens. Then you're going to look down the eyepiece. And then you're going to slowly move the stage away from the lens until the specimen is in focus. And then if you need to, you can swap to a higher objective lens magnification and refocus. So what's important here is that you're going to place the slide goes on the stage, then you select the lowest power objective lens, you move the objective lens to just below the stage, so using the coarse adjustment. Then you're going to look down the eyepiece lens, you're going to slowly move the stage away until it comes into focus, and then you can swap to a higher lens if you want to. Okay, so that's how we view it. The next thing that might happen is that we might then be able to see something. So we will see something a bit like this. So we're looking down there, we've got it in focus. These are perhaps what our onion cells are looking like. And something that we might be asked to do is to draw what these looks like. So when we're going to draw, we're going to be asked to do a scientific drawing. And we've got some rules. So we're going to use a pencil. We're only going to draw what we see, not what we think we see. We're going to use no colour or shading. Okay, we're going to go slightly over. We're going to use no colour or shading, and we're going to label with clear lines, and we're going to give the magnification. So I've got an example of that for you here. This is what that kind of drawing would look like. So these are your onion cells. You've got single lines. You're just representing where the different parts are. And you're doing really straight lines with direct labels on what you can see. So you'll be able to see the cell membrane, the nucleus, cytoplasm, cell wall. And then I've got the magnification labelled and I've stuck a title on there. So I'm going to stick that on somewhere as well. And I know we're over the 10 minutes, so I'm just going to put my title in here. And the last thing I'm going to recall as well is the final, the equation to calculate magnification and to calculate the size of the thing that you're looking at or the image that you're looking at. So we want to be really careful that magnification, so how many times bigger it is equal to image size. So image size would be the size in the actual image. So if we were looking at this cell from here to here, and you would measure that actually with a ruler, so that cell, the image size would be around 2.8 centimeters or 28 millimeters. That's your image size, and you're going to divide that by the real or the actual size. So the size that that cell actually is in real life will give you the magnification. So how many times bigger this cell is appearing to the cell as it is in real life. And that's an equation that you would be expected to use and to rearrange in your exam. So to rearrange, use it in different ways. So maybe you need to calculate the image size, maybe you need to calculate the real size, but you'll be using it in different ways. So you've got two kinds of methods. You've got information about the stains that you use, information about a light microscope. So this is a light one, not an electron one. How you would view a specimen, how you would draw what you see on the slide, and an equation that you would use to calculate the magnification. So I'm going to pop this video onto YouTube, and I've got some exam questions that go alongside. You'll be able to get them from the website.